Hi everyone, I'm Anahit. I'm a researcher from the Institute of Molecular Biology in Armenia, and I'm also visiting postdoc at the University of Cambridge. And I'm studying human populations based on the methods of evolutionary genomics. In particular, I'm interested in studying genetic history of Armenians based on both modern and ancient genomes coming from our region, from the Armenian highland. And I'm going to talk today about different types of data that is available in the field of genomics, as well as I will continue with what is now known about genetic history of Armenians. But I want to start with the basics in genetics because I understand that it might be the field you are not very much familiar with. So what is genome? You heard of DNA during your biology classes, or maybe we have biologists here, I hope so. Genome is basically it's a description of all the DNA we have in a living organism. And in all living organisms we have a DNA. It doesn't matter it's yeast or a fly, mouse, or human. So we have this molecule of life. And so back in 1953, when the double helix structure of DNA was discovered, and then our journey to understand this molecule of life began. And so now we know that inside of DNA there is a language, and which determines what kind of what we inherit, what genetic information we have, for example, what kind of diseases we may develop. And this language consists of just 4 chemicals. It's A, T, G, and C. So they stand for chemical entities or nucleotides. It's just 4 letters, but we have this kind of huge genetic variation in human populations that we see now. So it's in comparison, it's like 1 and 0 in computer science, right? And it's not less buggy, I should say. In human genome, we have about 3 billion base pairs, so it's quite a lot. And also one thing that you should know about genome that it is very complex, so it's more than genes, and we don't know entirely, we don't know the genome, like we didn't discover the whole code of life. And we call the major part of the genome that we do not understand the function of it, we call it junk DNA. But for sure in nature everything has its sense, so we're just on the journey to understand the meaning of this part of DNA. And one thing also that I want to make clear from the beginning of my presentation is that the number of genes in all humans is same. So it's about 25,000 genes in all of us, and there is no specific gene in a particular population. I don't know why, but in Armenia people think that we have so-called Armenian gene, or Hayugen, I don't know, probably you heard about this. So unfortunately there is no such gene, and there is no also Italian gene or French gene. Yeah, so bad news. How the genome look like? So it's like a long line of base pairs. So basically something like this with 4 letters, as I said. And back in 1919, there was an international initiative called Human Genome Project with the aim to sequence or to determine the whole genetic information in humans. So during more than 10 years, basically they did this. So they sequenced the whole genome, and it cost about $3 billion to do that. And I should say that the main breakthrough that helped to take off this project was the development of sequencing techniques recently, and now we were able to read genome fast, relatively fast, and relatively cheaper. So it costs now about $500. So you can compare $3 billion and $500. And now we know that all humans, we share about 99.9% of genetic information. So we have just tiny differences in our genome. And it's enough to have this broad genetic variation across human populations. So now the question, what we can do with all this genetic information that we know now? First, I should, of course, mention medical applications, right? So we are now moving towards personalized medicine. So it's when doctor may give you a prescription based on your genetic data. And so the second application, which I'm going to talk now about, is we can go deeper in our past. So I'm going now to talk about population genetics, the field I'm majoring in. And in a simple way, we can say that population genetics is a study of genetic variation between and within populations, and study of evolutionary forces that cause this genetic variation. So this is how now, today, genetics play an incredibly important role in understanding our origin, our evolution, migrations that took place across the world, and also to understand the relationship between human species. So this is how we know that in Europe there was two major migrations after the first Paleolithic migration. So it was a migration toward Europe from fertile Christians, of first farmers. It was around 7,000 years ago. And the third migration, it was a migration of Yamna people from steppe during the Bronze Age. And this is how we also know that ancient human, modern humans mixed with Neanderthals, so another species, sorry. And we know that in our genome we have around 1–2% of Neanderthal ancestry. This is also how we now know the story of Denisovans in Siberia, another human species. So if we talk about genetic markers used in population genetics, we can formally divide them into two groups. First group is uniparental markers, which means that they are transmitted solely from the one of our parents. We can talk here about Y chromosome, which is passed down from parental side, from father to son. And we can talk about mitochondrial DNA, which is transmitted from maternal lineage, yes, from our mother. And specific certain variants in Y chromosome or mitochondrial DNA, they determine so-called haplogroups or lineages or variants, or call whatever you want. And we can construct three of these lineages. As you can see here, this is lineages of Y chromosome. And of course, because humans, they originated or arose in Africa, so in the root we have African lineages. Now, each population has its own pattern of these lineages, of Y chromosome or mitochondrial lineages. And here on this slide, you see the typical Y chromosomal and mitochondrial DNA lineages for Armenian population. And of course, the major leap was with the development, as I told, it was the development of new molecular techniques like sequencing machines. And now we're also able to look at biparental genetic markers. This is a classical example in population genetics, which shows how genetic diversity is greatly mirrored by geography in Europe. And this is also how, based on biparental genetic markers, we know that in Chinese population of Han, there is about 4% of Armenian-like genetic source. And now, just in a nutshell, because it's a huge topic, I can talk a lot about ancient DNA, but this is a super powerful tool now in genomics because it changed the way we know, because it's greatly changed this field, because we can now directly investigate ancient samples, samples coming from our past. So what is ancient DNA? Ancient DNA is simply DNA which is extracted from ancient specimen. It can be bones, it can be teeth, it can be basically any ancient material like ancient hair or even ancient chewing gum. And so recently, Neolithic ancient chewing gum was discovered in Denmark, and the DNA preservation was so good that we can also reconstruct the girl who chewed this chewing gum. So she has a dark hair, blue eyes, something that is very different from what we see now in Europe, yeah? And even we can reconstruct with this method what she ate. So she ate dark and hazelnuts when she was chewing this chewing gum. Now shifting back to the Armenian genetics. So why it is important and why it is of interest to study Armenians. So it's basically not because we are Armenians, but there are some solid reasons for that. First, I should mention that we have a unique geographical position. So Armenian Highland sits northeast, north to the Fertile Crescent, which is an important region where agriculture originated. So, and also according to the many archaeological and historical studies, Armenian Highland play a key role in the migration, in the many prehistorical and historical migrations. Second reason is that we have strong ethnic and cultural identity. So we have our own alphabet, our own church, language, and our language originated approximately 5,000 years ago, so it separated from the main branch of Indo-European language family. And so basically these all factors they were conclusive for the genetic isolation of Armenians. And according to recent studies, so we genetically originated, formated about 4,000 years ago. And after that, we stayed genetically isolated. This genetic continuity was also backed up by studies of mitochondrial DNA, and we now know that in our region we were genetically isolated on maternal side, I should also mention this, about 8,000 years long. So this is really a lot for our region and in Europe, for Europe in general. One question that was less studied is regarding the origin of Armenians. And this is our recent study. So basically there are many theories, legends about formation of Armenian population, and in general we can divide them to two theories. So according to the broadly accepted Balkan theory, Armenians are not indigenous population. So they're coming from the Balkans because they were within the army of Phrygians coming about back in the Bronze Age. And according to the Herodotus, so it was his theory, proposed theory. So according to Herodotus, Armenian language also has similarities with the Balkan language. And they also had the same clause as Phrygian colonists coming from the Balkans. And the second theory is about local or indigenous origin of Armenians. And so using data from the region, using the modern and ancient samples, coming from Balkans and Armenian highland, we tried to check this Balkan theory. And so first we conducted a principal component analysis. Maybe you know about this analysis. It is very popular in population genetics. And here we see that modern Armenians, they are included in the cluster which contains also ancient samples from the region. And in stark contrast, we have Balkan samples in a separate quite far, as you can see here, cluster, which means that just looking at this plot, we see that we reject the Balkan theory. And then we conducted also a couple of analyses to support these results. So I'm not going to go deeper into the methods. I'll just say that we used these statistics to check whether Armenian samples, modern Armenian samples, they form clade with the exclusion of any other sample, like in this case, I was checking Balkan samples. And indeed, we see that, yes, Armenian samples, they form a clade, and no any other sample coming from the Balkans make any changes in this. Yes, good news. Okay, so then we also tested whether we have any other population except Balkans. And this is bad news a bit because, like, in this case, we found that there is a tiny, not tiny, about 20%, but still we have a genetic input coming from first farmers from the Near East. And this was around Bronze Age, so it was about 2,000 to 3,000 years ago. And so first conclusion here that the Balkan theory on the origin of Armenians is rejected based on the study of modern ancient data from the region. And then, so we have genetic continuity in the region, but we also found that there is a genetic input coming from fertile creation from first farmers, and the source of this genetic input is very similar to the one which was, again, at the same time back to Africa. So that means that we can conclude that there was a large-scale population movement across the Middle East around the Bronze Age. And this movement altered a bit modern Armenians from our ancestors. Of course, there are many questions. We still need to continue our studies because we need to understand where this migration was coming from exactly and what was the trigger of this population movement, whether it was climate changes or any other factors. We still don't know. Thank you. Any questions?
Those kind of models and which ones are preferred in genomic analysis? You mentioned PCA,
Yeah, I mentioned PCA. It's just a method to look in general on the genetic relationships. But we have, there are a lot of methods. I can talk a lot about Bayesian methods, ABC methods. So these statistics, as I mentioned, we can chat later if you want to learn more on these methods.
Yeah, thank you.
Thank you for an amazing presentation. Actually, I was kind of waiting for the results related to the Balkans example. My question is regarding the composition of the Armenian gen, and you said about the I'm not higher the Armenian gen. So the question is about the isolating. You said something like that our gen is isolated. I understand it that in the decomposition of it, there are no so many kind of nations in the decomposition of Armenian gen. So something like
I don't understand the question. Sorry.
I mean,
so
you said about
you want to ask about isolation.
Yeah.
This is the question.
And whether it has some weaknesses for the generation or something like that. I mean, when there is no many nations kind of merged with each other in the development of the gene during the centuries, so whether it has some weaknesses. Mm-hmm.
So, you know, from a medical perspective, that means that we we are isolated and we accumulate some diseases, yeah? So it's like same generations repeating every time. And in this case, I can mention maybe familiar Mediterranean fever, which is very frequent in our population. Why? Because again, there we have isolation, and actually every like there's a 20% of people who are carrying this mutation. It is again drawback of the isolation. So if you are talking about weakness, of course there are weaknesses in the medical perspective. I mean, we are accumulating some mutations. Yeah, let's say like that. Was it the question?
Yes, that was the question. So merging with other kind of nations have its advantages.
Well,
this is the kind of
the more diversity you have, the better it is. Yeah, in this perspective. Yeah.
Thank you.